to everyone for joining us today for the August session of the Excellence in Breeding GOBI webinar series. Again, my name is Kate and I'm from CIMMYT. But today we are very excited to have a joint presentation from two speakers who will talk to us about genomics data management and new BrAPI-enabled tools for marker-assisted breeding in Flapjack. Before we begin, I wanted to let everyone know that we will be recording this seminar and we'll make it available later for anyone who may want to access it. During the presentations, if you have any questions, please put them in the chat window. We'll be monitoring that, or you'll have a chance to ask them directly to the speakers when they finish. And before we proceed with the presentation, let's welcome our two speakers, Liz Jones and Iain Milne. I believe that many people attending this seminar have already had the good fortune to meet them, but we'd also like to introduce them to our new webinar participants. So first, let me begin with Elizabeth Jones, the current director of the Genomic Open Source Breeding Informatics Initiative project and the adoption lead for Module 5 of the Excellence in Breeding platform. Liz studied genetics as an undergraduate at Sheffield University and got her PhD in plant genetics from Springer University. She then did a postdoc at UC Davis, focusing on mapping QTL for late resistance in her first position after her PhD was with Agriculture Victoria, before she moved on to Pioneer, where she worked for 10 years on several topics, including setting up a high throughput genotyping. She next moved on to Syngenta for 3 years to lead a team focused on molecular marker assay development, and then joined the GOBI project based in Cornell in 2016. So please join me in welcoming Liz. And I'd also like to introduce Iain Milne, our second speaker today. Iain is the head of research computing at the James Hutton Institute, where he has worked since 2002, when it was still the Scottish Crop Research Institute. Iain originally studied computer science at the University of Dundee and stayed there for a PhD in software visualization too. He now splits his time between managing and maintaining Linux HPC resources for the UK's Crop Diversity Bioinformatics project, and developing informatics visualization and decision support tools such as Flapjack that he'll be covering today. So please join me in welcoming Iain too. And without further ado, let's proceed with the presentation. So Liz, the floor is yours.
Thank you, Kate, for that lovely introduction. And it's a great pleasure today to be able to share some of the GOBI achievements in the last 5 years. So GOBI stands for the Genomic Open Source Breeding Informatics Initiative. And this is a Bill and Melinda Gates Foundation funded project, and it's been funded for the last 5 years. We're just coming to the end of this phase of funding, and after this we'll be rolled into the Excellence in Breeding platform funding. So today I'm going to give you a short overview of the GOBI project, and then I'm also going to be showing you some of the latest tools that we've developed along with Iain Milne. So GOBI's mission is to enable the implementation of genomic and marker-assisted selection as part of routine breeding programs. And by routine, we need to be able to make decisions very quickly in time for the next breeding cycle. So we need to be able to load data quickly, extract and analyze data very quickly. And so that has been our key goals for this project. We are a global community. The core project is based at the Boyce-Thompson Institute, which is on the Cornell campus, but we collaborate with 3 different CGIRs with CIMMYT, IRI, and ICRISAT, and we have developers at those CGIRs, we have data curators who load data and test the system, and we have molecular breeders who bring us use cases for the breeding scenarios. And we also collaborate very closely with 2 different companies, Diversity Arrays Technology in Australia. They've developed our data QC tools, and the James Hutton Institute in Scotland, and they've developed our marker-assisted breeding tools in their tool, Flapjack. And we're very lucky today to have Iain to be able to demonstrate those tools. Okay, so what have we achieved in the last 5 years? Well, to begin with, we were working in year 1 on benchmarking different systems. Managing genotypic data can be quite challenging because of the dimension of the data. And so you need a very specific data management system to be able to handle loading, storage, and extract of that data. So we worked on building the basics of the system. Year 2, we worked on UIs for loading and extraction. And this was the first year that we worked with Flapjack and implemented the first tool and marker-assisted backcrossing. Year 3, we improved our permissions, delete and edit, and we worked with Dart on marker data QC and added pedigree verification to our Flapjack tools. In year 4, we worked on improving our overall processes. The system had got bigger than our processes. We implemented many new processes for QA, QC, automated all our regression testing, and improved our deployment. We also implemented genomic selection in Galaxy and forward breeding in Flapjack. So as we come to year 5, we have not added too much functionality to the core system. We have added tetraploids and we are adding indels, and we've added a very nice QC visualization tool with Dart. And in fact, Andrew demonstrated this tool at one of our last webinars. So the real focus of this year has been on integration, and that's what we're going to focus in this talk today. And we've made great progress in integration, and that's thanks to the BrAPI community. Just want to describe a bit about the BrAPI community. That stands for Breeding API, and API is Application Programming Interface. And the BrAPI community sets the standards for being able to exchange data between different systems and tools. And in the last year, they've made incredible progress on the genotyping data standards, and that has enabled us to have a step change in how we interact with other systems. So we've been testing out these BrAPI genotyping calls with various breeding use cases, and we can demonstrate a couple of those today. Also in the integration theme has been integration with the Excellence in Breeding Platforms enterprise breeding system, that we hope to take integration and analytics to the next level in the next phase of our project. Okay, I want to look a little bit about at the benchmarking that we've done on the system. To begin with, back in year 1, we benchmarked a number of different systems, and these are listed out on the x-axis here. And we had test data sets that we looked at the performance of these different systems. So our test data set was 5,000 samples and 32 million markers in this case. And you can see that the shortest time to load data was this HDF5 system here. And then I also want to look at extract times under 2 different scenarios. Here we see increasing number of samples with 32 million markers, and increasing number of markers in the millions with 5,000 samples. And with these increasing data set sizes in both scenarios, HDF5 extracted data the quickest under all use cases. So in fact, that's what we went forward with. We've developed an HDF5 based system that's extremely fast to load and extract. So when we started out working with the CGIRs, they had many different types of data. They were working with many different vendors, both external and internal, and many different formatting, data formatting types. So we've built a very flexible data loading system. We can load SNP data, codominant data, dominant data, SSR data. We started out focusing on diploid data, but now we have tetraploid data as well. And we can load different file formats, CSV, text, HapMap, and VCF. For the extract, we focused on just a couple of different formats. We have Flapjack to feed the downstream marker-assisted breeding tools. We have HapMap to feed the Galaxy genomic selection pipelines. And this is also a very useful format for a number of downstream pipelines. And then, as I said, we've really focused in this last year on BrAPI, on being able to pull data from the system using BrAPI calls and move it into other systems. So I just want to emphasize again that loading and extract is extremely fast for many different types of data. So this was loading data pre-COVID, pre-lockdown. I just want to show you how that's changed during lockdown. Moving on, just to check you're awake. Moving on, I'm now going to show you the marker data portal. So this is, once you enter into the GOBI system, this is what you will see. And I'm going to try and take us to a live version of the marker portal now. There we go. It's been a little slow this morning. Nope. Okay, seems to have got stuck. So now I am just going to take you by hand to that. That link doesn't seem to be working. So this marker portal is what you will see when you enter GOBI. So this is a live marker portal. I'm just going to feature a couple of the different tools that we have in the marker portal today, because I don't have time to go into all of them. So we have the data extract tool, and this was one of the first tools that we developed. And as I go into the data extract tool, I'll be asked to authenticate against the system. And I'm going to log in. And I can extract data by dataset, by samples, or by markers. And I'm just going to show you how to extract a dataset. Basically, just click on the dataset. If I want to see metadata about the dataset, I can pull up this little icon here to take a look at what's in that dataset. I can select my extract format over on the right here, either HapMap or Flapjack, and I can select a dataset and then just click Submit. Now, as I submit this, my data files will be extracted, and when they're finished being extracted, I'll be sent an email. And in the email will be a link to the files that I can then just download. So that's by dataset. As I look at by samples, I can filter based on various criteria. I can upload a list of samples, or I can copy and paste here. Same for markers. I can select a data type, upload a file, present a list, or I have this marker group feature here. And a marker group is basically a set of predefined markers. It can be markers underlying your QTL, or it can be markers that are used, for example, for purity analysis. So I'm just going to select that and extract. So that's a very quick overview of the data extractor. A quick look at our Galaxy tools. This is where our genomic selection tools are. You can see them over here. I'm not going to demonstrate these today. Stargau has given some very nice demonstrations of this at previous webinars. I just want to give a quick mention of the PedVer tool. I'm going to be showing you that later to be able to format data for Flapjack. And then as we look at our desktop applications, we can see Flapjack. And that's my cue now to hand over to Iain to be able to give you a demonstration of Flapjack. So I'm just going to stop sharing and hand this over to Iain.
Can you hear me and see that okay?
Yep.
Yeah.
Yes,
Yep.
thank you.
Okay. So this is Flapjack, which for those of you that might not know what it is, this is our desktop tool for doing graphical genotype visualization. It runs across Windows and Linux and the Mac. We also have a cut down version of it called Flapjack Bytes, which is linked to in the GOBI marker portal as well, which can be used to get little previews and snippets of genotyping data. If you actually want to get into the analysis side of things, then we use the full desktop tool. Now, what I'm going to do to begin with is just show you, haven't got time to do everything with Flapjack, obviously. I'm just going to pull in some sample data to give you an example of what it actually looks like to begin with. So the idea here is this is some SNP data, And we have a bunch of lines down the left-hand side here. I know you can't read the names just now, I'll zoom in just a moment. And then we have the markers listed across the top, or the SNPs listed across the top in map order. This is barley data. We have 7 chromosomes here. I can jump between them very rapidly. And then what the main display is showing is for each of the intersections, obviously between a line and a marker, we then have, I'll zoom in a little bit, we then have the allele calls. And where they are homozygous, we just represent that as an A or a G or a C or T. Obviously, this is nucleotide data. And where they're heterozygote, which I'll just highlight the heterozygotes, you can see we do this little split trick, so an A/G in this case. As I was saying, this is nucleotide data. We have lots of different color schemes for different data types and different ways of doing things. So just as a quick example, in this particular data set, let's say we wanted to color everything based on the similarity to that first line there. I'll do a genotype match on that. And that just confirms the line that I've picked. So that colors that line up in green, and then everything else where the alleles match that line, it obviously goes green as well, and where it differs, it goes in red. That doesn't change things too much, but if we then sort by the similarity to that line, let's put this again, so sort lines by similarity, and we can do this just for the current chromosome, or we can do this across all chromosomes. I'll just do it on this chromosome just now. Then what we should have, you can see in this little overview window down here, you can see obviously we have a lot more green towards the top of the screen where things are more similar to the line, and as we move down the chromosome, obviously there's a lot more red down at the bottom. This actually works really well. We've got some nice data sets in barley again with the cultivars, where you can scroll for miles across some of the chromosomes. You just have a sea of green where there's just a complete lack of genetic diversity in those regions. I'll flick back to the nucleotide color scheme for now. So what we've been working on closely with GOBI and partners at CIMMYT and IRI and ICRISAT over the last few years is adding a variety of analysis and decision support modules to Flapjack. We've been enhancing and extending the visualizations it supports, and doing things as well like just providing better support for data formats. So you can bring in Intertech data files, for instance, directly into Flapjack now. But before you get onto the data analysis side of things, you might want to clean up your data a little bit. So this could happen at the database side of things, it could happen at the Flapjack side of things. So you can see this data set here at the moment. We have a bunch of markers here where there's quite a bit of missing data. You can see there's 4 or 5 of them listed vertically there. So just as a quick example of the kinds of things you can do, we could go to edit and say we want to filter the markers and we want to filter missing markers. And you can do this in percentage-based data. I'll just keep the defaults here, whatever that's on. So get rid of all markers with greater than 30% of missing data, hit filter, and then you can see most of them have disappeared. So obviously there's still enough real data in this marker that it's kept that one behind. And one of the nice things in Flapjack is everything you do is undoable. So if you don't like that, I can just go up here and click undo the fact that it hits some markers and they come straight back. You can also mess around with the data in real time as well. So it's currently actually on what Flapjack calls as marker mode. I'm going to switch it to line mode, and I'll zoom in a little bit. And I'm going to highlight a line here as well. Let's see color scheme. I'll go back to similarity to this particular line. And I'm going to hold down the mouse here, and I can actually move this line around. So if I want to compare it to another line, you just sort of move it to a position where it's better to actually see that. This is all in real time. It's all very fast. Unfortunately, Zoom is probably going to destroy that for me in terms of if I start zooming in and out quickly and zipping around the data. Zoom will make it look like a dog swimming through treacle, but you just have to believe me that the tool does run really, really quickly. We can also do things like get visualizations of the actual chromosomes. This is not going to show up too well at all because this is very sparse data. Chromosome 3 here, you can see the distribution of the SNPs across that there. But most of the views are on this main genotype view, and that's where you'll spend most of your time looking at the data and analyzing the data. We can also bring in information to visualize QTLs and traits and display some heat maps and graphs and things based on that as well. Okay, so Liz mentioned BrAPI briefly, which is our common API that's been developed over the last few years to allow all of these tools across all of the institutes and all of the resources that we have to be able to communicate with each other without the user having to worry about it. So if you want to get data from GOBI or from Germinate or from GigWire or from SNP-seq into tools like Flapjack, then it should all happen automatically without the user having to worry too much about it. So to do that, I'm going to go to our import option. And we have various ways in Flapjack to pull data in terms of the example stuff I was just using, or map files and so on, but BrAPI has its own little option here. Quick little description of what it is. In the dialogue here, Liz will talk a bit more about how it works on the integration side of view later. on the integration side of things. So we just say we want to import from BrAPI, and what Flapjack does at this point is it connects to our server here in Dundee, and it gets a list of known BrAPI resources. Because we've been going through some big changes recently in the last BrAPI hackathon in Icrasat in February, everything switched to what's called BrAPI version 2, so we don't actually have that many that we know about at the moment. So the first one we present here is our Germinate Cactuar demo dataset. It's worth having a look and play around with this if you want to see how it works. But what I'm going to do for this example is override that and say, well, I've actually got a custom URL, which is GOBI's own one behind their firewall here. I'm going to connect to that. Pray to the gods of the internet that this is working. Yep, so we need to authenticate, so it's remembering my details, that's fine. And you have a few options in the BrAPI calls that it makes here to filter or change the data before we bring it back. So what I'll do here is tell it that I want to filter by study, which just makes sure that we're going to get some genotyping data in this case. And we won't bother with the maps in this particular example. So I'll click next again, it grabs that data. And then we have a list of all the information that it's pulled back over the BrAPI API from the GOBI system. And we're going to go for this last data set here, which is this little example one with some rice data. There isn't much information in the database about it, but if it had it, it would display it all here. And we click next again. And then so that's the studies that it had, and this is then what in BrAPI terminology we call the variant sets, which is basically our matrix of genotyped data that we want to get back. So you can see it's quite a small data set here. We've got just 11 lines by 9 markers. So this should come in quite quickly once we click next. And there we go, it's so small, I'm going to zoom in so we can actually see what we're dealing with here. So what we have here is a couple of parent lines up at the top here, these 2 lines here, and then a bunch of F1s after that. So what I'll do, and what we're talking about, is all the various analysis tools that we've added over the years. So we've got the marker-assisted backcrossing, we've got a couple of different ways of doing pedigree verification, we've got the forward breeding tool. So these are all available in Flapjack now, and the one we're finishing off just now, it should be available any day now, is a new version of the forward breeding tool called indexed forward breeding that we've been developing in collaboration with CIMMYT. But for this example, I'll show you how the pedigree verification tool works. You might want to run this one if, just to check your data and check that you've actually got a good cross. So we'll do the F1s because we know what the parents are in this case. So we have to tell Flapjack in this case what the parents are. There are ways of providing that information automatically to Flapjack as well, but this particular data set doesn't have that. And I think I can leave this on the default, so we'll just say that the first parent is this one, second parent is the second one there. If there were additional parental lines, we could say that we don't want to have them in the view, or the results view that we'll get, so we'll leave that one ticked. And we also want to simulate an F1 from the parent, so we can actually then compare the other lines to that expected F1 as well. We can, again, everything you do in Flapjack is all based on selections, so if we want to work across multiple chromosomes or even just multiple subsets of lines or markers, we can do that as well. So obviously there's so little data here, we'll just run across everything. And we can also pick some threshold settings. So this is where the decision support comes into it. I'll tweak these just back down a little bit. So this has various ways of then looking at this data once it's been analyzed, and Flapjack will tell you whether it thinks you've actually got a good F1 or not. So we'll leave this on the defaults for the analysis, and I'll show you how we can play around with that even after the analysis has been run. We'll run that. Again, it should be nice and quick. And what happens here, there's obviously a table of data displayed. But what we have on the left-hand side of Flapjack here, if you can see where my mouse is we have the 2 data sets that I've had loaded in so far. So I have this example data set and its default view, and this rice data set in the default view that it has. And we've now got an additional view has been created for this PEDVER F1's view 1. So you can see what it's done is it's created a new visualization based on this analysis, and the table is linked to that visualization. And by linked, what I mean is in the table, it's given us some analysis. So we have, I won't go too much into the statistics here. We've got obviously a bunch of columns. We've got the percentage of the data here. We've got heterozygous counts. We've got percentage heterozygous deviation from the expected F1s and so on. And the key thing is we have this selected column. So this is based on the decision that Flapjack has made. So it reckons in this case that this first of the non-parental lines is a true F1. It says that the second one is probably a self of parent 1 and so on. And it then selects the lines based on that. So what I'll do here is I'm going to untick some of these, and we'll see when we go back to the visualizations that more of these lines have been unticked because it's all linked together. If I then go back and select these ones and back to the table, then they should be selected again. And I can resort based in the table. Let's say we wanted to sort by the percentage of heterozygotes. That's okay. So we've now got 2102s at the top of the list. If we go back to the view, So 2102 is at the top of the list. Similarly, if I move this red line down to the bottom, that's 2106. And we go back to the table, and 2106 is down at the bottom. So it's all synchronized together. It means you can play around with the data either with the raw data that we have in this Excel style view or in the visualization itself. You can filter this data based on the criteria you can have. You can export it out to tools like Excel if you want to play around with it a bit more there. And as I was saying, so within the threshold settings, we can play around with this dynamically as well. So based on the parameters that you might want to input as we tweak these, hopefully you can see that the decision is actually changing in real time, and the selections that it makes are all dynamically based on that. So it's a really good tool for just playing around with that, seeing what the data works, seeing how the data looks compared to the visualizations, take it further downstream. And as I was saying, in this particular case, we want to find out whether you've got a good cross or not. So I'll leave it there for now and pass back to Liz. I'll remind myself how to stop sharing this one.
Okay, thank you, Iain. And I'm just going to back to my full screen mode. So Iain gave you a really nice demonstration of the basics of the tools that we have in place. But what we found were that breeders appreciated those tools, but they weren't necessarily using them on a routine basis. And when we surveyed breeders and molecular breeders, there were a number of different barriers to adopting those tools. The first one was easy integration. So you don't want to have to go to Flapjack extract, save your files, import them into Flapjack. You want to be in the Flapjack tool itself and be able to pull files directly from GOBI. And as Iain showed us, now we can do that with BrAPI. We don't have to be between two different tools. We can be in one tool and pull data directly in there. The second major issue, and this anyone that's worked in this space knows what a bear this is formatting data. So even though we have a Flapjack data export from GOBI that we can use directly in Flapjack, and we can pull a data set directly from GOBI into Flapjack using BrAPI, most of our genotyping projects are not one data set. They are multiple data sets. We might have 200 different F1 populations within a single genotyping project. And so to be able to have to split those into individual data sets is a huge burden for molecular breeders to have to do. And then of course you've got the parents that you need to have to do the pedigree verification. They may be in multiple places across that genotyping project and they may have multiple replicates. Now you have to pull all those together, either select a parent replicate to use or to do consensus calling. And by the time you've done all of that, of course, you may as well do all of your analysis in Excel. And that creates an awful lot of issues with introduction of errors and non-conformity and inconsistency of data analysis. So we've developed a tool now to help with formatting the data to deliver it to Flapjack, to be able to split data sets and do consensus calling in parents. Now you might have you've done all the preparation of your data, you've got 100 populations in Flapjack in different data sets. Previously you had to analyze each of those data sets separately, and now we have batch analysis in Flapjack that we can analyze all of those en masse. So this is what the process for the data formatting tool looks like. We pull data, again with BrAPI, directly from the GOBI database into this tool, and we can filter it. We do consensus calling for replicate samples, split the populations, create a Flapjack file, and analyze those in Flapjack. I'm going to see if my links work this time. Oh, there we go. So I'm going to go to my formatting tool, which is formatting data for F1 pedigree verification in this case. I'm just going to click on this. I'm going to select my study and my map set. I'm going to use a map set in this case. And this is a new feature. Now we can pull map sets with BrAPI as well. I can take a quick look at that data set to see I've got the right one. That looks very nice. Move on to the next step. I'm going to we just have basic filtering parameters right now. I'm just going to apply those filtering parameters, and I get a summary of what's happened after filtering. No markers were excluded, so I have 9 markers remaining, 105 sample, just 1 sample was excluded with that filtering. Again, I can check that data set by downloading, or I have a quick preview of the data set here. Now I'm going to do consensus building. I'm going to take a moment to just explain how the consensus calls are being built. We just have a very simple consensus calling method implemented so far. It's called majority genotype that favors the homozygotes. So for a majority genotype call, it's very simple. If you have 6 homozygous A's and 4 heterozygotes, ATs, for example, the consensus is going to be for the homozygous A. If you have equal numbers of homozygous A's and heterozygous, the consensus call is still going to be homozygous A in that case. It favors the homozygotes over the heterozygotes. I'm just going to apply that, and I can see in my tool immediately what the consensus calls were. So each of these are individual replicate samples, individual replicate parent samples, and this is the accession name for those replicates. And the consensus call is indicated with a star. You can see the star next to the accession name here. That indicates that that's being calculated based on the individual replicates. I can even go into the tool and edit the genotype if I want to. I think they look okay to me though. So I'm going to go to the next step and split the populations. Now, not everyone is entering their data into GOBI in a completely standard format across the CGIS. So we have a number of different ways that we can split those data sets based on what metadata is in the system. I know that my parents are unique to each of my data sets, so I'm going to split my data sets based on the combination of the 2 different parents. And I have at the bottom of my screen a summary of what was split. Now, one of those data sets didn't have any parent data, so we can't do pedigree verification in that case. But 4 of those data sets have parents, and we can proceed to analysis. So this last page is just a summary of what I've done. I'm going to download the Flapjack file, give it a moment to generate. And that's going to open up that project file in Flapjack. Okay, so now we're back to the view that Iain was showing you earlier. I'm just going to zoom in so we can take a look at each of the populations. But now what you can see on the left-hand side is that I have 4 different populations imported into Flapjack. And each of these populations has been arranged with the parents at the top, consensus called, and all of the F1s below. And now the magic of batch analysis. Before, Iain was using the single analysis. We're going to do a batch analysis and run this. And now for each of those 4 populations, I can see a decision on the right. I can adjust my thresholds if I'm not keen on, if I want to increase, if I want to select more of the F1s or less of the F1s. But now after batch analysis, I have a results summary, and this shows the 4 different populations and the proportion of F1s selected. You can see these populations were increasingly successful for the first versus the 4th, and then some average stats for each of those populations. Now, I would like to export these results. I can export only the selected lines or all of the lines. I'm just going to export those. I actually opened up this export file in Excel earlier on, so you can see what it looks like in Excel. So it's nicer formatting. And I have a list of all my lines across the 4 populations and the decision going forward on whether I'm going to make selections for those F1s. So this can be taken out to the field to make those selections. Okay, so that was batch analysis. I am going to just pause for a second and see if there's any questions on the batch analysis. There'll also be time at the end to be able to ask any questions. Okay, so I do have time. I wasn't sure whether I'd have time today, but I'm going to show you a new tool for forward breeding that's been brought to us from wheat use cases. From Umesh Rosara has brought us a new requirement for forward breeding that we just put into a beta version of a tool in Flapjack, and I'm just going to show you what that looks like. So I'm going to import a genotyping data set that's been prepared for forward breeding. And this is what the genotyping data set looks like. Now with forward breeding, I need to introduce a QTL file which has the favorable alleles defined. So I import the QTL file, favorable alleles are defined in this file. I have 7 QTLs. And then as I look at the default view, now I can see those QTLs displayed along the top of the genotyping data. My first QTL here has 4 markers underlying the QTL, and it's difficult for me to hover over the markers and the QTL at the same time. But you see those red lines coming down from the QTL that shows there's 4 markers underlying the QTL. I have 3 markers for this second QTL, and then I have 5 markers or QTL with 1 marker each. Now when I do my forward breeding, and this is a forward breeding tool that's already in the production version of Flapjack, and I run this, I go back to my breeding view. Now all of those alleles have been colored according to whether they have the favorable allele. So it's green for the favorable allele and red for the unfavorable allele in this case. And if I go to my forward breeding results, for each of these QTLs, I have a partial, like whether the haplotype has matched at a partial or a full level. And I've breeding view that all of my favorable lines with the most favorable alleles have come to the top. I can just deselect those I don't want and move forward with these selections. So that's our standard forward breeding analysis. Now to take this to the next level, I'm going to show you what the QTL file now looks like to be able to do a weighted forward breeding analysis, or what we call indexed forward breeding. So this was the format of the old file with a list of the QTLs, markers, and favorable alleles. And now we have the ability to return the trait value for those alleles. So breeders are often, especially for wheat, they're used to seeing the trait value rather than the individual allele. So we can return the trait value instead. And now we have a substitution effect for each of those QTLs and the relative weight. So this first QTL has a relative weight of 5 in this case, compared to 1 for some of the other QTLs. So this one's going to be weighted higher. So now when I go back and I can use the same set of data and the same QTL file. I'm going to do index forward breeding. Now what we see for each of those QTLs is the trait that's returned for those QTLs, and more importantly, a weighted molecular breeding value in this column here, which I can sort from largest to smallest. And now I can see that this sorting, it's also sorted the visualization. Any lines with this first QTL have been weighted at a higher level. So I can then remove any of those lines that I don't want and export these selections for further analysis. Okay, that's great. I managed to fit a demo of that new tool in. So this is what we're considering this a beta version of the index forward breeding. We're very interested in getting feedback from this community about how we can improve this and make this better. Okay, So now I am going to go back to my slideshow and just finish off with a look to the future. And so as we become integrated into the Excellence in Breeding platform, GOBI project is firmly embedded in Module 5, and this is bioinformatics, biometrics, and data management. And we're really excited about becoming integrated into their enterprise breeding system. And this will really take integration and data analytics to that next level. For the GOBI tools, these are all publicly available. These are all the links that will be shared to this community that you can access all of these tools. We do have a deployable version of the system now that you can access from this link, or you can just ask us for a deployment and we can send that to you and help you get your data loaded. So I want to finish off with thank yous. I want to first of all thank the BrAPI community. This is a community with representation from germplasm banks, international consortiums, private companies, public institutions, crop-specific tools. And this is a very important community because they are setting the standards for being able to exchange data between different systems and tools. And the progress that they've made in this last year in the genotyping space has been really fantastic. And I want to give a shout out to Pete Selby, who coordinates this community and is funded by Excellence in Breeding to do this. And that's really enabled us to implement a step change in integration and being able to share data between systems and tools. This is a very big slide, so it might take me a little while to get to the next one. Here we go. So I do want to also give a very special thank you to each of our steering teams. We have steering teams at each of the CGR centers that we work with, and our scientific advisory board with representation from academia and also private industry. And we have a fantastic group of Ithaca PIs who have been very, very much part of the process of developing GOBI and very vested in our future. And last but not least, I want to thank all of the GOBI team who have been a fantastic team to work with, very engaged. This has been a real pleasure to work on this project. I want to give a special shout out to the people that developed the tools we demonstrated today. That's Angel and Vishnu, who developed the PedVer data preparation tools. And then Umesh and Carlos and Star were very instrumental in giving feedback to the Flapjack tools. And the James Hutton Institute, of course, developed the tools, and that's Iain Milne and Gordon Stephen. So thank you very much, and we'll take any questions.
Thank you very much to Liz and to Iain for the great presentation. And thank you to everyone who has already put some questions into the chat. I believe several of the questions have already been answered, for example, about tools that are available for GWAS and GS analysis. There have been responses from Star and Juan Burgueño, who's also based at CIMMYT and has collaborated on the GOBI project about the availability of those tools with Tassel and Galaxy and some tools that are available in Dataverse. So if you have questions about GWAS or GS, you can consult the chat box, but we can also answer questions about that as well. But there are some questions that I think still remain open in the chat box. So maybe just starting from the bottom, and then I will go up. One question from Damian Platten was how you handle non-reference haplotypes within a QTL interval. So Damian, I don't know if you'd like to say anything else about that question.
I can if you wish. Do you understand the question though?
I think so. You're saying, so if you have a specific haplotype underlying a QTL, but you have something that doesn't quite match that, how do you handle the trait returned?
Yeah, so in your QTL definitions, you've got a definition of your favorable favorable alleles at a set of markers, right? Mm-hmm. Now that will span a significant genetic distance in many cases. Some cases it won't, but in many cases it will. And so you will end up with situations where you have non-reference haplotypes, so haplotypes that don't match the donor or the unfavorable, so the favorable or the unfavorable haplotypes in that interval. And that could be due to recombination if it's a large interval, or it could be just simply due to breakdown of LD, or those particular markers are not in perfect LD with the QTL. So there's a number of ways that can arise. But how do you handle that in the analysis?
It's a really good question, and thank you for asking that, Damien. So we have a couple of different options that we have in place now, but we're really looking to improve this and would really appreciate your input on this. The first option is we do like a percent. So say this is the haplotype ATTG, we do a percent of match. So if it's ATTC, then it would be 75% match for that haplotype. Another option that we have is to be able to say, well, which is the key marker that we really need to be able to have to make a decision on whether that's a favorable allele or a non-favorable allele. So to define the markers that we have to have under that haplotype. Now, we don't have the best way to be able to do an array in this table, but we would like to implement, I think, which is more your use case, Damien, where you define all of the different allelic combinations and given it determining whether it's favorable or unfavorable and what the trait determination is based on that array of different haplotypes. Does that answer your question?
Yeah, sure. Thanks.
Okay.
Thank you, Damien and Liz. Another question that came in towards the end of the presentation was from Alaku on what is the optimal number of SNP markers for PEDVER?
It depends on the crop. I would say, we have very good success with PEDVER with 12 to 20 markers. You could certainly have more, and if you have enough definition of those markers, then you could have a few less as well. We do have some tools in the Galaxy pipeline to be able to help you select the best markers for variety ID or for pedigree verification. But in terms of Flapjack tools, Flapjack can handle hundreds, thousands of markers. But for pedigree verification specifically, we're looking at just small numbers of markers, 10 or 20.
And on that topic, in terms of size, Noelle from SIP was just asking if there are any limitations on the data frame size in terms of samples or markers.
Yeah, I mean, if you're talking about Flapjack specifically, then it's really just about how much memory you have. So I think out of the box, Flapjack defaults itself to about 4 gigs, which is not that much on a modern PC. And even with that amount, it can happily handle, a few million markers by thousands of lines. I mean, obviously, as you change the dimensions, you can have it really, really wide or really, really deep, or somewhere in between. It's difficult to define a sort of fixed limit on that. Increase the memory, the data sizes go up. So you can throw it at clusters with tens of millions if you want.
Noelle, do you have any specific data set size that you're considering that you'd like to ask about?
Hi, yeah, thanks. No, I think that answers it. We just have, collection-wide data, which is usually, anywhere from 4,000 to 6,000 samples by, 50, 60,000 up to 100,000 markers. Depending on which genotyping platform we use. So just several things that we've run into in the past where that it was just really hard to find different programs that could handle that type of data frame and be able to process it. Especially because we don't have, good access to a cluster computer or something like that. So that's why I was asking. Thank you.
You might have to bump the memory limits up on Flapjack a little bit, but I think you should be able to cope with that.
Cool. Thank you very much.
Thank you. So another important practical question that came in about being able to use the tool for the F1 verification is how you associate the F1s with specific parents. So if you're going to do that analysis. So maybe Liz, if you could describe that.
I think this why I had this slide open. So what we do when we are doing an F1 pedigree verification is we have the parents defined, and then we simulate an F1 based on the parents. So if the parents are AC, we expect the F1 to be a het AC match. And then we just compare each of the F1 lines to that simulated F1. So for example, this line here doesn't match that expected F1. It in fact does match the parent, so it's likely to be a self. I don't know whether that helps with the question. Did that answer it?
I think I meant actually more technical, how the software knows to connect F1/1 to parents P1, P2. How that information, the pedigree information is stored.
I see. So that is stored in the database, what the parentage is.
Okay, I see, thanks.
So those are metadata associated with the samples in GOBI Live, is that correct?
Right now, yes. In the future, that could be metadata associated with those same samples in the breeding management system. But right now, we have that information stored in GOBI.
Thank you. Okay, sorry, I'm just checking. So there's another question that just came in, if there's any software for doing duplicate analysis in large germplasm collections of gene banks or breeding germplasm collections.
So from the GOBI tools that have been developed, you could probably do a sort. We're talking about each germplasm collection having one sample or multiple samples?
Here in the germ plant, this Rajnish who asked the question. My question is, suppose we are doing the collection from the different countries. So maybe the same accession in the other countries with other name, but now we have the molecular tools, we genotyped it. Now maybe the line is the same, but it has the different name. So, and do you have any software who suggests, no, these are the 5 lines from the different country, but they are the same lines?
Yeah, I think that would be fairly straightforward to do in Flapjack tools. You could just do a sort or a genetic distance analysis to be able to group germplasms collected in different countries that could be the same.
Okay. No any other method or something for particular this, except this cluster analysis? Because generally cluster analysis is the, I mean, normal people use, but any other sophisticated method or something?
Not a specific tool to be able to do that, but I think, you know, that would be fairly easy. I'm sure Iain could collect your requirements and be able to put a tool together, fairly easy to do that, because it's basically a sort and similarity of alleles.
So how much do you have, could you give the how the use threshold and other the sequencing error? How to do that? Do you have any experience on that? Because now we don't know that this issue is coming, we are thinking of the sequencing error, or it's really something threshold. Getting the threshold is a big issue in this correction. So sometimes I think maybe you have a software, you know, the software will take care for and make, okay, this is that threshold, you can use something. That's why I asked that.
Yeah, and I think that's very important to be able to collect replicate samples of each of the germplasms you're looking at, so you can see the sequencing error from those replicates and set a suitable threshold. It's very important.
Okay, thank you.
Thank you. So going up a little bit in the questions, there was one relatively early on from Gary Atlin, who had asked about how GOBI functionalities would be accessed within the EBS.
Right, and we hope that a lot of these tools will be available through the analytics pipeline. We could see, you know, I know we've talked about this, Kate, that you could just have the analysis output directly through the analysis pipeline, and then you could click on that and then open up Flapjack if you wanted to get a visualization of that. That's how I see it anyway.
Thank you. And I also wanted to bring.
This is
Sorry.
Tom. This is
Hi, Tom.
Can people hear me? I mean, hello. I mean, technically, as we connect, it's the EBS service gateway, which I won't go into any details on, but that's how parts of the system, GOBI database, would connect to that. and then a tool like Flapjack would connect to that. Or as Liz mentioned, by providing data from breeding system GOBI database to the analytics framework, that's where you bring data together for analyses. So it's through the service gateway how different parts of the system connect. So you don't have to rely on all data being breeding data is in the breeding system, genotype data is in the GOBI database, and through the service gateway, you bring those information together and provide it to things connected to the service gateway.
Thank you. Okay, so we also had a question from Mary, and some of her questions were answered, but I think one of the outstanding ones was how to perform and interpret LD analysis with genomic data without information on marker positions. So Liz or Ian or others, would you like to comment on that question?
Ian, I think you commented on that one in the chat.
Ah.
Do you want to answer that one?
Sorry, I was typing messages to somebody else. Which question?
So on doing an LD analysis with genomic data without information on markers. And I see, yeah, now also that Liz mentioned that there's Tassel tools.
Yeah, I mean, I think my comment was just that we would like to try and add something like that to Flapjack. We've been talking about it for years, but my understanding of the LD side of things is that it gets more and more difficult from a visualization point of view as we have more and more lines. So there could be a screen resolution challenge to that, but certainly on the analysis side of things, I don't see why we couldn't do something.
And there's also tools in Tassel to do this. And Star was just saying that Tassel is also incorporated into Galaxy as well. So there's pipelines in Galaxy to do this.
Great, thank you. So I think we only have a few minutes left, but does anyone have any other questions that you would like to ask our speakers today? Okay, or if I missed any questions in chat, please feel free to also bring them up.
Yeah, Huang has a couple comments. Katie, you want to read out on the chat?
Or maybe Juan, would you like to just make provide the information you were giving on the gene bank analysis and other topics?
No, just I put two main comments. One about the distance matrix that Flapjack is calculating, because we need to take care if we use it, because when you use the distance item, you have a match distance. But if you are using clustering, the program is using a different distance, I guess Euclidean distance. This is one of the comments. And the other is about the discussion of duplicates, because in the work we are doing with Noel and the other gene banks describing the variability between and within accession, we found that it is very, there are a large variability in the results depending on the species, the level of allogamia of the species, and other kind of thing. For example, it is clear in maize, if we are comparing CMLs, lines that breeding lines, you cannot use the same threshold to identify duplicates than if you are comparing accession or wild relatives. This is an important issue that we need to consider when we are comparing results. Also, it will be useful if we can have a kind of p-value or a better statistic to compare when we are comparing as duplicate accession, or maybe also if we are comparing the F1s. That's the number of matches are not enough to declare some ones as different with the parents or not.
Thank you, Juan. Do we have any other last questions from anybody on the webinar? Okay, if not, then please join me in thanking very much.
Last one, Kate.
Oh,
yeah.
Sorry. So the last question that I just saw pop up is it possible to access the data from R? Is the question from GOBI to be able to pull it directly into R? Is that the question?
There's a fantastic BrAPI API for R that exists that you'd be able to use to do that.
Okay,
Excellent.
thank you.
Okay, well, thank you very much. And please join me in thanking our wonderful seminar speakers today for telling us about these tools. And thank you to all of you for participating, for your active engagement. And we hope that you can join us for the next seminar in September.
Thank you, Kate. Bye, everyone.
Bye.
Bye.
Thank you.